we have the student and the staff on show.
Yes. Good morning, Dr. Dien. How are you?
Yes, I'm good, Tanto.
That's good. That's great to
Yes, long
hear.
time no see.
Yeah, long time, very long time. Okay. So I think Prof. Helmi have a trouble to join. Hmm, wait a moment.
Okay, good morning, everyone. Ladies and gentlemen, and distinguished guests, respected lecturers, and dear students. It is a great honor and pleasure for me to welcome all of you to today's guest lecture on basic knowledge of virus, bacteria, and fungi from Dr. Dien. First and foremost, let us express our gratitude to God Almighty for granting us health and the opportunity to gather here today. I would like to extend my warmest thanks to all of you for your presence and enthusiasm. Before we proceed, let me briefly outline today's agenda. First, Welcoming speech from Dr. Wiwi. Next, documentation. And the third is session to moderator. And the fourth is presentation. 5 is question and answer. 7 is closing the agenda. Before we start the agenda, let us pray together. Pray begin. Pray has ended. Okay, the next agenda is welcoming speech from Dr. Wiwi. Hello, good morning, Dr. Wiwi. Can you hear me?
Okay, yes, good morning. Thank you. Sorry, I'm on the way. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning to all. The Honorable Vice Dean of Faculty of Veterinary Medicine, Universitas Erlangga, the Honorable our special speaker, Dr. Dien. I'm sorry if I'm mispronounce your name, Doc. All division veterinary microbiology lecture and to all the students. On behalf of Faculty of Veterinary Medicine Universitas Erlangga, it is my great pleasure to welcome you all to this guest lecture event. This guest lecture has become an annual lecture event for veterinary student semester. The objective of this lecture is to update and increase the knowledge about microbiology science from international experts. Today, we invite Dr. Thanh, lecturer at Faculty of Applied Technology, School of Technology, Van Lang University, Vietnam. Dr. Lee talk about basic knowledge of virus, bacteria, and fungi. We hope through this lecture, students can increase understanding of microorganisms, including virus, bacteria, and fungal cells, as a basic for learning virology, bacteriology, and mycology. So, on behalf of Faculty of Veterinary Medicine, Universitas Erlangga, we would like to express our thanks to Dr. Lee for sharing your knowledge. I think it's enough. Once again, welcome to event. And finally, we hope that all the participants could follow this lecture. Wabillahi taufik wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh. Back to MC. Thank you.
Okay, thank you for Dr. Wiwi for welcoming speech. The next agenda is documentation for the committee to take role for documentation. Please, for all the participants, can you turn on the camera, please? We wait until 10:15.
Recording in progress.
Okay, for the committee, please take the documentation, please. Okay, I will count from 1,
Recording
2,
stopped.
3 for slide 1. Next to the slide second. 1, 2, 3. Next slide. 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 1, 2, 3. And the last slide. 1, 2, 3. Okay, thank you. Okay, the next agenda is the main session for Dr. Lee Tan Dien and will be given to the moderator.
Okay.
For Dr. Ruri, time is yours.
Okay. Thank you very much, Sarah, as a MC today. Okay. Good morning, everyone. Could you hear me? Yeah. Okay. First of all, I would like to say thank you very much. It is such honor for me to have the opportunity to introduce our speaker and guide us through this exciting lecture that organized by Microbiology Division, Faculty of Veterinary Medicine, Universitas Erlangga. So today we are going to have a guest lecture with Dr. Lee Tan Dien as a guest speaker, and he will talk about basic knowledge of virus, bacteria, and fungi. And before we begin, Let me introduce our guest speaker first. So, Dr. Lee Dien, he's graduated PhD from Chulalongkorn University, Thailand, and right now he is a lecturer at Faculty of Applied Technology, School of Technology, Phan Lang University, Vietnam. And he has many experience and research in the field of microbiology. Okay, I would like to greet Dr. Lee Dien first. Maybe I can call Dr. Lee. Hello, Dr. Dien. Dr.
Hello.
Dien, are you already with us?
Yes. Yes. Hello.
Okay, great. Thank you very much, Dr. Dien. Let me give your information that your talk will be around 45 minutes and followed by the discussion session around 15 minutes. And also information for all the audience during the guest lecture session, you can ask a question by tap your question in the chat box. And if you want to ask question directly to the Dr. Dien, you can use the feature raise hand in the Zoom meeting in the discussion sessions. Okay, all right, everyone. Please welcome our guest speaker, Dr. Lee Dien, with the topic Basic Knowledge of Virus, Bacteria, and Fungi. Dr. Lee Dien, time is yours.
Okay, thank you so much. First of all, I would like to thank Dr. Wiwi for the welcome speech. Thanks Elangard University, the Faculty of Veterinary Medicine for invitation for the guest lectures from the lecturers in Vietnam. So thank you so much for your introduction, Dr. Ruri. Yes, and firstly, I would like to introduce a little bit about my research. Actually, I read the PhD from the Faculty of Veterinary Science and Chulalongkorn University in Bangkok, Thailand. Yes, my research is focused on antimicrobial resistance and development of the alternatives to antibiotics to combat with the AMR, especially in the bacterial infection in livestock industry, especially in aquaculture. So for today, actually we have senior lecturers, professor, and students from Erlanger University. So the topic is the basic knowledge of the virus, bacteria, and fungi. However, I just have you to remind very briefly about the knowledge of virus, bacteria, and fungi. After that, I have maybe a half of the section to share about experience to work with the bacterial phase. So let us start my presentation.
Recording in progress.
Can you see my slide now?
Yeah,
we
we
can
could
see
see
your
your
slides.
It's a full screen now, right?
Yeah, it's already full screen.
Okay. Okay, let's start the section for today. So as I mentioned before, today's session is the basic knowledge about a virus, bacteria, and fungi. So I keep the section at the first part of the very basic knowledge about the microbiology, including bacteria, yeast, and fungi. And the second part of the lectures, I would like to focus on bacterial phase and the application of bacterial phase in veterinary medicine, especially in livestock industry and aquaculture. So for the first part is the fundamentals of microbiology. We know microorganism is diversity in the nature. So we can know about bacteria, yeast, fungi, and virus, and easily to look at the pictures with the different size of the bacteria. virus or other organism, we can see the nano size. That means the very small organism, coronavirus or bacteriophage, they have nano size maybe less than 100 nanometer. It's very small compared with the red blood cell, especially in the bacteria such as Salmonella or E. coli. So we can know a microorganism, it defines a living thing that is so small. It must be viewed with the microscope. However, with the virus and bacteriophage is a nano size, so we cannot use the normal microscope. We have to use transmission electron microscope to look at the morphology of the virus. So for the other like bacteria or a beta cell, we have to use the normal, the conventional microscope to look at the morphology of them. So based on the structure, we can divide the microorganism into two groups. The first one is acellular, and the representative of the acellular microorganism is virus. And the second group is cellular microorganism. So for the cellular microorganism, we can divide into two groups, prokaryotic and eukaryotic. For the prokaryotic, we have the bacteria and archaea. And for the eukaryotic, we have angle, protozoa, and fungi. So when we look at the fundamental of microbiology, it's very diverse in the natures. We usually think about the microorganism with the disease, like the bacterial disease or viral disease or fungal disease. However, a lot of useful microorganism in the natures. So we will take a little bit time to discover about the useful microorganism very soon. So I have you to remind a little bit knowledge about the bacteria. So the morphology of the bacteria we can divide into two categories. The first one is cocci, the second one is bacilli, and the other set of the bacteria we can set into the third category. For the cocci bacteria, they can arrange as the coccus, diphylococci, or streptococci. We can see the very common bacteria like Streptococcus thermophilus as a useful bacteria for yogurt fermentation. They have a Streptococci for the morphology. And for the bacilli, we usually think about the short rows bacteria. However, sometimes we will see the long rows bacteria or Coccobacilli bacteria. And in the screen, we can see the morphology of the Clostridium botulinum and Vibrio cholerae. It's a very dangerous bacteria. They have a risk for food safety. For the bacterial cell structures, I think we know before about the structures of the bacterial cells. Usually, the bacteria contains the genetic material, DNA, circular DNA, or DNA in plasmid, ribosomes, pili, cytoplasm, plasma membrane, cell walls, capsules, and some bacteria they have the bacterial phlegm. So for the bacterial cell walls, we usually divide the bacteria into two groups, the gram-positive bacteria and gram-negative bacteria. And the difference between the RAM positive and RAM negative is basically based on the peptidoglycans. So for the positive RAM positive bacteria, we have the thick layers of peptidoglycan compared with the RAM negative bacteria. So we can differentiate RAM positive bacteria and RAM negative bacteria by RAM staining. So the function of the bacterial cell walls usually to provide the cell with a structure support, shape, protection, and the function as a selective barrier. Moreover, the bacterial cell wall can have the cell withstand the osmotic pressures and the mechanical stress from the culture's media. So I would like to mention about the bacterial cell membrane. So we know about the cell membrane of the bacteria is a phospholipid layers. So in the surface of the phospholipid layer membrane, they have the proteins, maybe lipoprotein or lipopolyprotein. So the cell membrane can have to provide a protective barrier around the cell and regulates which material can pass in or out the cell membrane. So about the bacterial spore, spore is a small oval or spherical structure that are very resistant to the high temperature, radiation, desiccation, and chemical agents. It is very important for the bacteria that can tolerant in the high temperatures or in the high level of the chemical agents. So we can see the spore of the bacteria can locate in many positions, such as in the center of the cell, with the representative is the Bacillus anthracis. This is bacteria can cause the anthrax disease. It's very dangerous. And the second one, the spore located in the terminals of the bacteria cell, with the representative is the Clostridium tetani. And another is a Clostridium botulinum. They have the spores in the subterminal of the cell. So for the real life application of the bacteria, when we see the bacteria, maybe we see about the bacterial disease, the infectious disease, very dangerous. However, a lot of useful bacteria we can use in the real life. For the example, we can use the bacteria as a very important items to research about microbiology, to research about the mechanisms of drug, the interaction between drug and the bacteria, can have to understand and have to develop the drug or other type of the treatment. For the second one, maybe we use the bacteria for nitrogen fixation for agriculture industry. Especially we can use a biofertilizer. We can use the bacteria for bioremediation for the toxic chemicals in soils or sediment. And especially the bacteria can apply for the medicine industry, especially pharmaceutical, like antibiotics, or we can use the bacteria for the cosmetic or the medicine to treat or to improve the health of the human. So continue with the fungi. So fungi is popular in the nature also. So morphologically, fungi can categorize into four. The first one is yeast form. The second one is yeast-like form. Continue with the moss or filamentous fungi and dimorphic fungi. For four of the fungi, I think it's a very important, very popular yeast and filamentous fungi. So I will give you some basic information about the yeast and fungi. For the yeast morphology, we can see many types of the morphology of yeast. However, the popular morphology is a round, spherical, elongated, broad, and apiculate, oblate of the shape. So we can see the size of yeast is bigger compared with the bacteria, especially virus. So they can use the nutrition in the culture medium to grow rapidly compared to the bacteria. So the colony of yeast usually bigger compared to the bacteria colony. For the yeast cell structure, it's a little bit different compared to the bacteria. As I mentioned before, the cell walls of the bacteria contain the very important components is the peptidoglycan. However, with the yeast cell structures, the most important components is glucan, especially beta 1,3 and beta 1,6 glucan. So we can differentiate between the yeast and the bacteria by the size. When we see under the microscope, it's very difficult to observe the bacterial cells under the microscope. However, we can easily to see the yeast cells under the microscope. And based on the cell walls component, we have to remember the difference between the components of the cell wall of bacteria and yeast. Bacteria is beta-glucan is important, but the yeast is beta-glucan. Actually, the components of the cell walls of yeast, including chitin also, and some monoproteins. However, the most important component of the yeast is beta-glucan. And when we think about yeast, maybe we think about the application of yeast in the food technology. However, we have many aspects of the yeast application in the real life. First one is fermented foods. I mean, fermentation technology that use yeast as the agent for wine production, beer production, alcohol production, or pig production. However, we can use yeast to be an agent for biodegradation of the toxic chemical in the environment also. Yeast is rich in beta-glucans in the cell walls. So beta-glucans in immunostimulants are very good for human and animal to increase the immune system. So the yeast proteins provide a high qualities. It one of the alternative proteins for to tackle the problem of the bombing population around the world. So when the population increase, we have to put a pressure to feed all of the people. So we have to find a new source of the protein, replace for meat, especially chicken, pork, or beef. So many researchers, they have to find a new solution such as a single cell protein from bacteria, fungi, or yeast. Some solution they suggest that insect and other type of protein from aquatic animal is very important. However, in the case of yeast, they have a lot of application in food technologies, especially for the high level of protein, high level and high quality of protein source. So continue with the filamentous fungi. When we see the pictures in the screen, usually we think about the fungi, especially in filamentous fungi. Filamentous fungi is easy to differentiate between this type of microorganism with the others, because we can see by the naked eye. So about the colony morphology of filamentous fungi, for the first one, the fungi usually use spore for the raw forming, and after 24 or 48 hours of the cultures with a suitable medium, the fungi can grow, and we can see its naked eye very easy. So in the slide, we can see some type of the fungi, especially I would like to mention here is a Aspergillus flowers. Aspergillus flowers is very popular fungi. So the fungi, they can generate the aflatoxins, a very dangerous toxics. They can cause a harmful for the human health and animal health also. So usually we think about fungi, we think about the color, but actually the fungi, they don't have the color. The color comes from the spore, the fungal spore. So in case of Aspergillus flowers, we have the yellow spore. So when we look at the morphologies of the Aspergillus flowers, we can see the yellow color. So for the structures of the filamentous fungi, usually we have the hyphae. The hyphae is a long filamentous structures that form together as a network. We can call this a mycelium. And some of the fungi, they have the rhizoid. And the important component of the fungi, we can see the sporidium. The sporidium is the structure of enclosed units with spores are formed inside. And the spores is the very important component of the fungi. It's a biological particle which fungi make for reproduction. Many types of the reproduction styles of the fungi, however, usually they use spore. In the screens, some structures of the filamentous fungi. You can see the mycelium, the hyphae of the fungi, and the cell wall of the fungi. So for the cell wall, the fungal cell wall, the most important components is chitin. Like we mentioned this before, the bacterial component, the important component of bacteria is bacterioglycans in the cell wall. With the yeast, beta-glucan, and the fungi, the most important is chitin. So even though they have the protein and the leucins, especially beta-glucans in the cell walls of the fungi, however, the most important component is chitin. So for the application of the fungi, maybe we think about the toxicity of the fungi. It's a very harmful for the human health. However, the filamentous fungi, they have a wide range of application in the real life. In this slide, I would like to focus on the application of the fungal enzyme. The fungi is one of the more important microorganisms that can generate the enzyme. Enzyme is very important to apply in the food technology or other type of environmental treatment solution. They can apply in industrial field, environmental field, and biomedical field. So for the industrial field, we have a symbol like cellulose. Cellulose is the enzyme that can degrade the cellulose, so we can apply in the food tech we can think about the betinase. Betinase is an enzyme that can degrade the betin. Betin is a important component when we use the fruit as a raw material for wine fermentation. Because for the fruit, they have high level of betin. So betin is a viscous and they can reduce the juice used for the fermentation technology. So if we apply the betinase, they can degrade the betins and increase the concentration, increase the use of the fruit juice, so they can affect for the fermentation technology. Other type of the enzyme like proteinase, they can degrade the proteins, silase, or many kinds of extracellular enzyme. They can use the enzyme to mix with the as a feed additive for animal, even though for the human. So now I go to the fundamental of the virus. I mentioned before, virus is a nanosize. So that means we cannot see the virus by the colony like bacteria or yeast, and we cannot see very clearly like filamentous fungi. We have to use the transmission electron microscope to look at the morphology of the virus. So actually we have many ways to to classify the virus. However, usually we can divide the virus into 4 groups. The first one is helical virus, such as the tobacco mosaic virus. It is a very serious viral disease in tobacco. The second group is polyheterovirus with the adenovirus. The third group is the cervical virus. We can see is the influenza virus belong to this group. And the last one is complex virus. And the representative of the complex virus is bacteriophage. When we think about a virus, we think it's a nanosize. It's very, very small. And we think about the infectious disease like coronavirus, HIV, SBV, or rabies. So it's a very dangerous. However, with the bacterial phase, it is youthful virus. So because I worked on bacterial phase for many years, so today I would like to share with you a potential application of the bacterial phase in vet medicine, especially in livestock industry, including poultry, pig farming, or aquaculture. So the bacterial phase, or we can call it phages, is a special virus that can kill the bacteria, they infect the bacteria and kill the bacteria. So in the pictures, we can see the bacterial cells in green and the purple is the bacterial phase. Usually when we got a bacterial infection, we use antibiotics. However, the misuse of antibiotic can induce the antimicrobial resistance. So antimicrobial resistance increase year by years and a dangerous situation for the human health and for animal health also. So many centers, they have to develop the new solution for the bacterial infection treatment. Like we can use the phytochemical, that's been the herbal extraction. We can use robotics. We can use the nanomaterials. However, for prevention of the bacterial infection, we can use the vaccination. Vaccination is very important for the human health. However, in my research field, I focus on the bacterial phase. So basically about the structure of the bacterial phase, you can see it's on the screen. They have three parts. The first part is the heat. Outside of the heat is the major capsid protein, and inside of the heat is a genetic material, maybe DNA or RNA, based on the type of the virus, based on the type of the bacteriophage. Some of the bacteriophage, they have the color, and the others a non-color. And the last part of the bacteriophage is tail. A tail very important for the bacteriophage because they have a long tail or a short tail. However, they have the protein of the tail. Protein of the tail is very important because it can absorb the cell bacteria by the receptor, the specific receptor in the cell membrane, in the cell wall. So when we look at the pictures of transmission electron microscope image of the bacteriophage T4, we can see with the same with the 2D or 3D pictures in the lab. So for the bacteriophage, we can divide into two groups: the lysogenic bacteriophage and the lytic bacteriophage. I mean, lytic bacteriophage, that's it, the virus, they can infect and kill the bacteria. However, in case of lysogenic bacteriophage, when they observe the bacterial cell and pump the genetic material inside the cell, the genetic material merge with the bacterial genetic material, so they cannot kill the bacteria. So for the application of the bacteriophage, we focus on lytic bacteriophage. For the lytic circle bacteriophage, they have 4 steps. The first step is absorption. The second step is penetration. That means the bacteriophage pump the genetic material inside the bacterial cells and left the body outside. And the third step is the replication and maturation. That means the bacteriophage will generate the component of the phage body inside the bacteria. After that, assemble together to make a new bacteriophage inside the bacteria. The last step is the release. That means the bacteriophage will induce the protein. That's protein they can degrade the cell membrane and cell wall. So inside the bacteria have a lot of the bacteriophage, the new bacteriophage, maybe 100 or 200. Some bacteriophage, they can generate 50 or 80 the new bacteriophage inside the bacteria based on the type of the bacteria and type of the bacteriophage. So very important for the absorption. If the bacteriophage cannot observe in the surface of the bacteria, they cannot kill the bacteria. So which factors are the most important for the absorption of the bacteriophage and the bacteria? This is the protein in the tail of the bacteriophage and the receptor in the surface of the bacteria. They have to link together with the specific mechanism. So in the pictures on the right hand side, is the bacteriophage specific for Edwardsella etaloris. It is the bacterial can cause the disease in many types of freshwater fish like tilapia or catfish. So as I mentioned before, the bacteriophage is a virus, so we cannot see the colony of the bacteriophage in the petri dish. However, we have the specific technique to look at the black, I mean the clear zones of the bacteriophage. If they have the bacteriophage can infect and kill the bacteria, so they can form the clear black on the petri dish. We can see the clear black here. It's very clearly. So for the application of the bacteriophage in vet medicines, I have to suggest the roadmap for the phage research and application. So we would like to apply the bacteriophage as a biocontrol agent to control the bacteria concentration to kill the bacteria to protect our animals. So for the application, we have to do many steps. Work with the bacteriophage is not difficult, however, a little bit complicated because we have to conduct a lot of steps for isolation, characterization, purification, and application. So firstly, we can think about the bacteriophage. If you would like to do with the bacteriophage, we have to isolate the bacteria first. So it means we have to our host bacteria is the food for the bacteriophage. So for the livestock industry, maybe aquaculture, poultry, or pigs, a lot of bacterial infection in the livestock industry. Maybe it come from E. coli, Salmonella, Campylobacter jejuni, or in aquaculture we have Aeromonas hydrophila, Edwardsella etheluri, or many types of the bacteria infection in livestock industry. So first one, to start the research and application, we have to go for the sampling. Sampling, that means we collect the sample from the animal, and after that we do the bacterial isolation. After we have the pure colony of the bacteria, we go for the bacterial identification. So for the bacterial identification, we have many types of the methods to do the identification. Conventional methods, we use the biochemical testing, or we use the DNA-based methods, as mean the PCR and sequencing, or we can use the protein-based methods. We can use the MANDI-TOF MS. MANDI-TOF is rapidly and they can generate the result within 10 minutes after we have the sample preparation. So especially very difficult to differentiate between some type of bacteria. They have the similar biochemical characteristics. So for the most exact methods that used for identification based on the whole genome sequencing. So after we have the collection of the bacteria, we can do the antimicrobial susceptibility testing to identify multi-rate resistant bacteria or the normal bacteria. After that, we do the pathogenicity testing to identify the virulence of the bacteria. After that, we have to conduct the experiments to identify the candidates of the host that used for the phage isolation. So I mentioned before, we would like to focus on lytic bacteriophage. That means the bacteriophage can infect and then can kill bacteria. So if you would like to isolate the lytic bacteriophage, we have to check the bacterial host. They did not contain the raw phage inside. Raw phage, that means the lysogenic bacteriophage. If they have the lysogenic bacteriophage inside the bacteria, I mean, when we do the technique to for phage isolation, maybe this is a lysogenic bacteriophage, not lytic bacteriophage. So if the bacterial host don't contain the prophage inside the bacteria, this is the most potential candidate for phage isolation. We have the technique for phage isolation. We can call this the double layer agar technique or black assay technique. So after we have the phage strain and phage collection, we have to do the biological characterization of the bacterial phage by chemistry and electron microscope to check the morphology of the phage. And we have to do the whole genome sequencing to identify exactly the family of the bacterial phage. After finish this step, we have the lytic bacteriophage stain, and we can go more for the sampling to collect a sample for phage collection. So after we have a lot of strain of the phage collection, we can make the lytic phage biobank. So phage and bacteria are very specific. So maybe the bacterial phage isolated from Vietnam cannot apply in Indonesia. So we focus on indigenous bacterial phage. That means if you would like to apply in Indonesia, maybe you have to work with the indigenous sample, like go to the farm, collect the sample, collect the animal, collect the environmental sample like water or soils or sediment to try to find the local bacterial phage for the application. So after we have the lytic phage biobank, we can do the customized phage cocktail. Phage cocktails, what's it? Phage cocktail. Phage cocktail is mixtures of the bacterial phage. I mean, when we mix the bacterial phage strains together, we can control the co-infection of the bacteria infection in the animal. Like in poultry, we usually know about the co-infection of the E. coli, Salmonella, and Campylobacter. So if we have the bacterial phase, we can mix it together to make a product, and we apply the product to reduce the concentration of the E. coli, Salmonella, and Campylobacter in one product and one time. So this is the suggested roadmap for phage research and application. So I will give you a example based on my research. The first one is the using lytic bacteriophage against the pathogenous multi-drug resistant Aeromonad heterophyllus in tilapia. So firstly, we have to do the Aeromonad heterophyllus isolation and identification. We do the AST test, virulent assay, profile induction to choose the most potential candidates multi-drug resistant Aeromonad heterophyllus. And I go for the phage isolation and characterization. After that, I do the efficacy testing in the wet lab to check the efficacy, the protection efficacy of the bacterial phage when the tilapia are challenged with the multi-drug resistant Aeromonad heterophyllus. So I will show you some results. In the left-hand side is the bacterial phage black. When we see on the petri dish, and the figure B is the morphology of the bacteriophage under TEM, transmission electron microscope. And when I do the challenge test and efficacy test in the wet lab, we can see is that compares to the control. That means the challenge with bacteria and without a phage therapy, the phage group that can increase the RBS of the animal experimental animal RBF from 50 to 73.3% as a very potential for application. And the second one, I would like to give you the example of the isolation and characterization of the bacteriophage infecting multi-drug resistant Edwardsella ictalorhiza in Bangladesh catfish. So we go for the farming for the sampling, and we isolate the bacteria, the ictalorhiza, Edwardsella ictalorhiza, inside the body, internal organs of the Bangladesh catfish, do the bacterial isolation. Yes, and for the phage isolation, you can see it's on the pictures with a clear black, very big black and clear. So that means the phage is a very strong activity that can infect and kill a lot of bacteria. So for this research, I will show you a video for to easy to look at the results and the research. So I share with you the video. Sorry, they have me check the video on the screen, right?
Yeah, yes, we can see, but
You can hear the voice
no,
of the
no.
video.
No, we cannot hear the video.
Okay, maybe it's Vietnamese, so you can look at the subtitle. Yes.
Yeah, all right. Dr. Tin, you would like to try play the video?
Yeah,
Because we cannot see the video
you
on the
cannot see the
screen.
video.
Yeah.
Okay, let me check.
Thank you.
You can see my screen now.
Yes, we can see now clearly. Thank you.
Okay. Uh, this is the example of the phage applications in our culture. However, we can apply the bacteriophage in other aspects of the vet medicines as the poultry industries or the pig farming. So another project, we focus on the E. coli, the multi-drug resistant E. coli in poultry. Especially we think about the colistin resistant E. coli. So we can use the bacteriophage to control the multi-drug resistant E. coli. So when we have the result, we can suggest that E. coli lytic bacteriophage a potential for the phage cocktail, and the phage cocktails a potential to apply to reduce the concentration of the bacteria in the animal. So as I mentioned before, this one is the suggested roadmap for the phase research and application. If you would like to apply the phase in vet medicine, you have to do the many steps. It's not difficult, however, a little bit complicated, just step by step to do the research and apply. So the bacterial phase is the potential solution to tackle the antimicrobial resistance in human health and even though in the animal health. So this is my university, the campus, the main campus of my university. So I will finish my presentation for today, and I welcome the question and the sharing from the professor and the student. We can discuss, we can talk, and we can change the knowledge together. Thank you so much.
Okay, thank you very much, Dr. Dien, for your wonderful and inspiring presentation. And now we are moving to Q&A session or discussion sessions. Is there any questions to Dr. Dien about his talk today? So maybe while waiting the question from the students or the others, maybe I would like to ask to you, Dr. Dien. So I'm quite interested about bacteriophage. Yeah, bacteriophage because that you mentioned you said that misuse for by antibiotics is
Yes.
yeah, it will be a problem about antimicrobial resistance. So I would like to ask about, you already mentioned about the advantage of the bacteriophage in the animal health, but could you explain to us what is the challenges between we use bacteriophage or we use traditional antibiotics treatments? Thank you.
Okay. Thank you so much, Rui, for your question. So I will explain about the phage applications in vet medicine. Actually, the bacteriophage is a virus. So if you would like to research about the virus, we have to have a very good facility, right?
Yeah.
However, with the bacteriophage, it's very simple.
Mm.
I mean, we no need to use the cell culture system because the bacteriophage infect the bacteria. So we have the biosafety cabinets and we culture the bacteria. And after that, we do some step for the isolation. It's not difficult to research to do with the bacteriophage. And no need to use the high quality of the facilities. So it's very easy to do if you have to work with the bacteriophage. And because we would like to apply the bacteriophage in the farm, so the bacteriophage is a virus, it's a live microorganism. So we have to pass and to get a certificate from the government.
Mm.
So I don't know the regulation, the national regulation in Indonesia about the use of the live microorganism in farming. However, in Vietnam, when we use the probiotics, because probiotics is the live microorganism also, and we have the criteria, and all of the company have to submit the document about the detailed descriptions about the products and about the risk to spread of the infection of the products in the farm. So if we have the regulation to use the live micro organism in the farm, it is very easy to apply the bacteriophage. And the bacteriophage we can generate with a high concentration. So basically, we culture the bacteria after 24 hours or 36 hours. We have the concentration is about 10 power 8 or 10 power 9, very high, right? However, with the bacteriophage, we can generate 10 power 11 or 10 power 12 of the phage particle in 1 milliliters. So if we would like to apply in the aquacultures, it's a little bit difficult because in the commercial ponds of aquaculture or the cake cultures, we have a large of the volume of the water. So if we would like to apply, we have to dilute the bacteriophage too much. However, if we apply in the hatcheries or the closed system, it's more easier than with the commercial pond. For the livestock industry like poultry or pig farming, it's more easy to apply the bacteriophage. We can use the bacteriophage powder or the liquid of the bacteriophage and put directly in the water. After that, we can feed the water for the animal. And the concentration to use in the animal like chickens or pig is low. Maybe many researchers suggest the concentration for the application is 10 power 6 or 10 power 7. So we can generate the product with 10 power 10, 10 power 11. So with a high concentration. So it's very easy to apply in the poultry industry or pig farming. And because the bacteriophage is a special virus of bacteria. So they infect the bacteria, kill bacteria, and they didn't kill the probiotics or the microbiome inside the intestines system of the animal because it's very specific. That means the bacteriophage of E. coli, they can infect and kill E. coli. They cannot infect and kill Salmonella. When we apply together with probiotics, they could not infect and kill our probiotics. So it's very specific. So that's some advantages of the bacteriophage for apply in the animal health. However, we have to face with some disadvantages because bacteriophage and bacteria interactions is specific. So I mean the geographical distribution. That means the bacteria from the north is different from the south. It's very diverse in the nature. So maybe the bacteriophage in the north cannot apply for the south, and the phase in the Indonesia cannot apply in Vietnam. So we have to check about the host strains, about the infect activity of the bacteriophage before we apply in the farm. So the first one is the diversity of the bacteria in nature. The second one's because the bacteriophage and the bacteria interaction in nature is co-revolution together. So based on this characterization, the bacteria have to develop the mechanism to resist to the bacteriophage. So maybe for the long term of application, the bacteria resist the bacteriophage like antimicrobial resistance. However, because the genome size of the bacteriophage is very small compared with the genome size of the bacteria, so the speed of the revolution of the bacteriophage is more faster than the bacteria. So when the bacteria resists to the bacteriophage will develop the mechanism to adapt to the change of the bacteria. So the last ones, because the bacteriophage is not news for the human health. However, we need to wait the approval from FDA or from WHO for application. But we can start with the research first. After that, we have the collection of the bacteriophage we keep in the biobank. And after we have the approval from the organization, we have the material to go for the application in the field. So the very good step to repair before we wait until the approval of WHO or FDA. These are some points I would like to mention with you about the potential application of the bacteriophage. And even though in the veterinary medicine, a lot of bacterial infection, right? And almost they use the vaccination for prevention and use the antibiotics for treatment. Yes. and we use a lot of antibiotics. They can increase the antimicrobial resistance, not for the animal health, for the public health also. So we're very dangerous, very serious nowadays. So I don't know the situation in Indonesia, but in Vietnam, next year, antibiotics will be not used as the role promoters in the livestock industry. That means you have to use a treatment, and we have to decryption from the veterinarian before we use the antibiotics in livestock industry. And in Vietnam, we have the national regulation about the use of antibiotics in aquaculture onshore. However, we have to face with the problem because with the big company, it's easy to control. However, with the small farm, they don't care about the regulation. So when the disease outbreak, they would like to protect the animal first. So that's why they would like to apply the antibiotics. And antibiotics is easy to buy in Vietnam now, and support from the company, the commercial company. So that's why we have to think about the sustainable solution to reduce the use of antibiotic and reduce the AMR in the future.
Okay, Dr. Dien, thank you very much for your alignment informations. That's quite interesting. So besides the advantage using the bacteriophage for replication or substitutes using the antibiotic treatments, but we also have challenges for using the bacteriophage. Okay, thank you very much for explanations. So maybe there is a question from the students from Nashwa Tasi. Okay, thanks for the